Hello, students. This is Dr. Kelly Sexton again, and this is Respiration Part 3. In the last video, we looked at gases and we looked at ventilation. Remember, ventilation is moving air into and out of the respiratory tract. When we move air into and out of the respiratory tract, air fills the lungs. Well, we can measure this movement. This is what's called a spirograph or a pneumograph. And if you look at this, you'll see several different volumes. So when you look at it, you'll see just normal, quiet breathing. This looks like an up and down wave, sort of like the tide going in and out. This is called the tidal volume. So tidal volume is the amount of air that we move in a normal breath. But we can move more air if we wish. We can breathe in more. That's the inspiratory reserve volume. We can breathe out more. That's the expiratory reserve volume. If we add the three of those together, that's a maximum breath in and a maximum breath out. That's called the vital capacity. But notice down at the bottom of this graph, the green part. This is air in the lungs that cannot be breathed out. Remember, our lungs are always partially inflated because of the lower pressure around the lungs. This air that cannot be breathed out is called the residual volume. And so if we want to know the total lung capacity, we have to include that. If we look at respiratory volumes, we can use those to assess a person's respiratory status. If you look at an average adult male and you look at an average adult female, all the numbers are different. Males in general are bigger than females. They have bigger chests, and so they have more capacity. The only thing that is different or not different is the tidal volume. And if you notice, the tidal volume for a male and the tidal volume for a female are basically exactly the same, 500 milliliters. Everything else, like I said, is different. You should be able to interpret this graph. You should be able to label the different parts of the spirograph or pneumograph. However, you don't have to know the individual amounts or volumes. These are just averages. I would, however, like for you to remember the tidal volume. Well, of the air that comes in, not all of it makes it down to where we get gas exchange. A lot of air is included in the nose, the nasal cavity, the pharynx, the larynx, the trachea, the bronchi, and so on. That air has no gas exchange, and sometimes it's called anatomical dead space. If we're going to talk about the air we're exchanging with, we have to subtract this anatomical dead space. So with the average breath being 500 mils, anatomical dead space of 150 mils, only about 350 milliliters of air is exchanged. Another thing about the air is it changes as you move down. When air enters your nose, it's mostly oxygen and nitrogen. But as you move down, it mixes with the air from the previous breath. So by the time you get down into the alveoli, there's much, much more carbon dioxide, much, much more water vapor, and less oxygen and nitrogen. This is because of the humidification of the air, the mixing of the old air with new air, and also gas exchanges that happen in the lungs. Look at this graph. It's a comparison of the partial pressures of these gases in atmospheric air and alveolar air. We already know that the partial pressure of nitrogen is about 597 millimeters of mercury. Remember, it's 78.6% of the air. We know that partial pressure of oxygen is about 159 millimeters of mercury. Remember, it's 20.9% of the air. We know that carbon dioxide is about 0.3 millimeters of mercury. It's about 0.04, 0.038% of the air. If we add all of that up, we get about 760. Also notice that there's not a lot of water in the air that we breathe in. If we then look at the composition of the air down in the alveolus, it's very, very different. Look what happened with the water vapor. It went from 3.7% or 3.7 millimeters of mercury all the way to 47 millimeters of mercury. It went from about 0.46% to about 6%. Look at what happened to carbon dioxide. It goes way up. It goes from 0.3 all the way to 40 millimeters of mercury. It went from about 0.04% up to about 5%. That means these others have to go down. So oxygen in the air we breathe has a partial pressure of about 159 or 20.9%. But by the time you get down into the lungs, it's dropped to about 104 or only about 13% of the air that's down there. Nitrogen also falls, but remember nitrogen is fairly inert to the lung and we don't really need to worry about it very much. The two gases we're going to spend most of our time talking about are oxygen and carbon dioxide. Again, partial pressure of oxygen in the air that we breathe in, about 159. Partial pressure of oxygen in the alveolus, about 104. Partial pressure of carbon dioxide, the air that we breathe in, about 0.3. Partial pressure of carbon dioxide in the alveolus, about 40. Well, this partial pressure down in the alveoli is the air that we're doing gas exchange with, not the air that we breathe in our nose. And so if you look at inspired air versus alveolar air, they're quite different. Remember, as the deeper you go, we're mixing it with the air from the previous breath. Conversely, when you head in the other direction, you're mixing it with air that has not had gas exchange. In other words, air with more and more oxygen, less and less carbon dioxide. So if you look at expired air, there's quite a bit more oxygen than what's down in the alveoli. There's less carbon dioxide than what's down in the alveolus. Remember, the only one of these that we're doing gas exchange with is the air that's in the alveoli. And so the absolute maximum amount of oxygen that we could possibly get into our lungs is no more than 104 millimeters of mercury of oxygen. The absolute lowest we can get carbon dioxide out of our blood is no lower than 40 millimeters of mercury. So we're going to look at respiration. Well, first we're going to look at external respiration. External respiration is gas exchange between air and blood. And then we'll come back later and look at internal respiration, which is gas exchange between blood and the cells. Remember, in both cases, it's going to work based solely on diffusion. And diffusion only works from higher concentration to lower concentration. Diffusion stops when equilibrium is reached. So when we look at external and internal respiration, we have to take into account these partial pressures of the oxygen in the air, in the blood, and in the tissues. Let's look at external respiration. Again, it's gas exchange between air and blood. So air comes in to the alveolus, and then blood comes. It's already been out to the tissues. It's low in oxygen, high in carbon dioxide, sort of this bluish color, and we're going to get gas exchange. Again, gas exchange happens based on diffusion. There's more oxygen in the alveolus, so it will enter the blood. There's more carbon dioxide in the capillary, so it will enter the air. Well, if we look at this, we can see exactly what's happening. So look up at the very top at atmospheric air. Remember, here's the partial pressure of oxygen, about 159, 160. Here's the partial pressure of carbon dioxide, about 0.3, 0.4. That air changes dramatically as it enters the respiratory tract. Remember, we're going to clean it, we're going to hydrate it, and we're going to mix it with air from the previous breath. So by the time it gets into the alveoli, the partial pressure of oxygen is going to be much lower, about 104, 105. Partial pressure of carbon dioxide, much higher at about 40. Again, this is the gases we're exchanging with. And then if you look at the blood on the left side, it's coming from the right side of the heart. The right side of the heart receives blood from all of the systemic tissues. It's low in oxygen and higher in carbon dioxide. And if we look at the levels, the carbon dioxide is about 45. Oxygen is about 40. When it reaches the lungs, we're going to get gas exchange. There's more oxygen in the alveolus. It will enter the blood. There's more carbon dioxide in the capillary. It will enter the air. And by the time the blood leaves the lung, it's going to look almost identical to what we saw in the alveolus. Maybe not exactly, but almost identical. So the partial pressure of oxygen is going to go way up to about 100 or so. Partial pressure of carbon dioxide is going to drop from 40 down to about 45 down to about 40 or so. That blood is now brighter red, high oxygen, low carbon dioxide. It's going to go to the left side of the heart. And then the left side of the heart, when it contracts, will pump the blood out to the tissues. So we should remember what these numbers are. Partial pressure of oxygen in the alveolus, about 104, 105. Partial pressure of oxygen in the blood, about 40. It's going to go from high concentration to low concentration. Well, it happens fairly rapidly. It only takes about a third or so of the time that the red blood cell is in the capillary in order for this to happen. And so if we look at the time in the capillary, you can see exactly what's going on. At the start of the capillary, we're low oxygen. The blood is bluish. But very quickly, as it moves through the capillary, we're going to reach maximum about 100 millimeters of mercury oxygen. We're going to reach that very quickly. In about a third of the time it takes to get through the capillary, we're going to completely finish this gas exchange. The same thing's true for carbon dioxide. Although it has a lower partial pressure gradient, which means it's not going to move quite as fast, it's about 20 times more soluble in water or plasma than oxygen is. And so it's going to diffuse in an equal amount. So if we look at those pressures, the partial pressure of carbon dioxide in the blood is about 45. Partial pressure of carbon dioxide in the alveolus is about 40. It's going to move from high to low, so it's going to go from venous blood into the alveolar air. Don't forget, it's going to move in equal amounts with oxygen, but that's only because it's much, much more soluble than oxygen is. Blood is then going to move through this circulatory system, and we're going to wind up at the capillaries in the tissues, and we're going to get another gas exchange. This time, though, the gas exchange is between blood and tissues. Again, this is called internal respiration. So internal respiration is gas exchange between blood and tissues. It's very similar to external respiration. It always moves from high concentration to low concentration. So again, we need to pay attention to what these concentrations are. If we look at the concentration of oxygen in the blood, remember it's been to the left side of the heart, it's pumped through the aorta, out through the arteries, And now to the capillaries. It has a high partial pressure of oxygen and a low partial pressure of carbon dioxide. The tissues, on the other hand, are using the oxygen and they're producing carbon dioxide. And so they're going to have a much lower partial pressure of oxygen and a higher partial pressure of carbon dioxide. So we're going to get gas exchange. Oxygen, because it's higher in blood, is going to move from blood to the tissues. Carbon dioxide, because it's higher in tissues, is going to move from the tissues into the blood. And by the time the blood leaves these capillaries, it's going to look very, very similar to what we saw in the tissue cells. The partial pressure of oxygen will be about 40. The partial pressure of carbon dioxide will be about 45. That blood is then going to return to the right side of the heart. And then it's going to be pumped to the lungs, and we're going to have external respiration again. So if we look at this, again, it's gas exchange between blood and tissues. The partial pressures here are reversed. The diffusion gradients are reversed. Oxygen is higher in blood, and so it diffuses into the tissues. Carbon dioxide is higher in the tissues, and so it diffuses into the blood. So it's a circle. So blood, as it circulates, that's again called a circle, we're going to see a constant external respiration followed by internal respiration, followed by external respiration, followed by internal respiration. Again, it works completely on diffusion, and so diffusion only works from high concentration to low concentration. So in the lungs, oxygen is higher in the alveolus, 105. It's lower in the capillary of the lung, about 40. Oxygen moves from air to blood. Carbon dioxide is higher in the blood than it is in the alveolus, 40 and 45. So it's going to move from blood to air. When we get around, we're going to have internal respiration. Now the oxygen is higher in the blood than it is in the tissues. So it's going to move from blood to tissues. Carbon dioxide is higher in the tissues than it is in the blood. So it's going to move from blood to tissues. And then the whole process is going to repeat itself over and over and over again. If I were you, I would pause and take a couple of minutes and look at this. Here's inspired air, alveolar air. You can see the partial pressures of oxygen and carbon dioxide in the blood, in the alveolus, and in the tissues. Find the path and talk about with yourself what's going on at each place. We're going to have external respiration at the lungs, internal respiration at the tissues. Well, that means that oxygen had to be transported. It's transported in the blood, and it's transported two different ways. One way is attached to hemoglobin. If you remember from blood, red blood cells are packed full of hemoglobin. There's about 287 million molecules of hemoglobin in one red blood cell. And all that hemoglobin can attach to oxygen. Remember, you can attach one oxygen per iron. There are 4 irons in each hemoglobin. So we can carry 4 oxygens per hemoglobin. So most, about 98.5%, is attached to hemoglobin. A very small amount is just dissolved in the water of the plasma, only about 1.5%. This is a picture of the hemoglobin molecule. You can see we have the globin part, that's those peptide chains. We have these 4 hemes. In the center of the heme is the iron, and that's where the oxygen attaches. 1 oxygen per iron, 4 hemes per hemoglobin, 4 oxygens per hemoglobin. Well, in the presence of oxygen, it attaches to hemoglobin. But when we reach an area where oxygen is low, it comes off. this oxygen binds loosely and reversibly. When it's bound, we call it oxyhemoglobin. When it comes off, the hemoglobin is then called deoxyhemoglobin, or sometimes reduced hemoglobin. If we move back to an area where there's more oxygen, it's going to pick it up and turn into oxyhemoglobin. If we move back to an area where there's low oxygen, it's going to come off and turn into deoxyhemoglobin. There's an equilibrium established. So the more oxygen there is, the more oxygen binds to hemoglobin. The less oxygen there is, the more the oxygen comes off. So in lungs, we have more oxygen, so we have more oxyhemoglobin. In the tissues, there's less oxygen, and so the oxygen comes off, and so we have more deoxyhemoglobin. This adding and coming off is called loading and unloading. So we load oxygen in the lungs and we unload oxygen in the tissues. Well, when we do this, we can graph what's going on, but we don't get a straight line. The reason why is because affinity, or how much hemoglobin likes oxygen, changes. As oxygen binds, 1, 2, 3, 4 oxygens, as they bind, hemoglobin likes oxygen more and more. The affinity goes up. And then as hemoglobin comes off, affinity goes down. So we don't get a straight line. Remember, oxygen doesn't all have to be bound to hemoglobin. Hemoglobin can carry up to 4. So when it's fully saturated, it has 4 oxygens. Partially saturated when it only has 1, 2, or 3 oxygens. If we graph this, we can see it. We can also see it in a figure like this. This shows oxygen from the alveolus entering the blood. Most of the oxygen is going to enter the red blood cell. It's going to combine with oxygen or hemoglobin and give us oxyhemoglobin. Remember, a small amount is going to be carried dissolved in plasma. The only reason this happens is because of diffusion. There's more oxygen in the alveolus than there is in blood. When we get to the tissues, exactly the opposite thing happens. There's more oxygen in the blood than there is in the tissues. And so the oxygen moves down its gradient. So the oxygen that was dissolved in plasma comes out, and then the oxygen that was bound to hemoglobin releases and then moves from the blood to the tissues. When we get to the lungs, the exact opposite happens. There's more oxygen in the lung than there is in the blood. Oxygen enters. When we get to the tissues, exact opposite thing happens. So remember, this is called external respiration, and this is called internal respiration. And just like we saw before, it happens over and over and over and over again. Again, we can graph this oxygen bound to hemoglobin. So if you look at this graph, on the left, you'll see what it says, percent oxygen saturation. In other words, how much of the hemoglobin has oxygen attached to it? 0, 20, 40, 60, 80, 100%. And if you look across the bottom, you'll see the amount of oxygen, the partial pressure of oxygen, 0, 20, 40, 60, 80, 100. Well, remember, in the alveolus, the highest we get is about 100. In the tissues, we go down to about 40 at rest. Well, look at this graph. So this graph shows what happens as we provide more and more oxygen. It binds to hemoglobin, but it's not a straight line. If it were a straight line, follow the line out until we reach 100%. That's at the far right of this dotted line. If you come down, look how much oxygen you would require to reach 100%. Now, it's not listed on this graph, but you can estimate it. It would probably be about 120 to 130 millimeters of mercury, and it's never that high in the lungs. So if this were a straight line, we would never be able to fill up our hemoglobin to 100%. But because this is not a straight line, look, it fills it up right around 100. So this property that hemoglobin has makes it much, much more efficient at picking up and dropping off oxygen than it would be if it did not have this change in affinity. Another thing is, as oxygen travels through the capillaries, not all of the oxygen comes off. At rest, only about 20 to 25% of oxygen comes off. What this means is if you start exercising, if oxygen levels in the tissues begin to drop, there's plenty more oxygen, about 75 to 80% of the oxygen. We did not drop it off before, but now that extra oxygen begins to dissociate from the hemoglobin and it can be used by the cells. In other words, we have more oxygen to give to our tissues without increasing respiration rate or cardiac output. So if you look at this graph again, remember, lungs, we have a partial pressure of oxygen about 100. Tissues, we have a partial pressure of oxygen about 40. Well, look at the curve. It drops off from 100. It keeps dropping off down to 40. But look, there's still somewhere around 75% of the oxygen bound to hemoglobin. We still have 75% of available oxygen that was not dropped off. Again, if those cells, those tissues need it, we have all that extra oxygen to provide. We can't really change the shape of this graph. This solid line does not change. It has a sort of S-shaped or sigmoid shape. We can, however, shift the curve. We can shift the curve to the left or shift the curve to the right. When shifting, what happens is the affinity changes. If you shift the curve to the left, affinity goes up. In other words, hemoglobin likes oxygen more, which means it grabs it easier. It's easier to pick it up. On the other hand, if we shift to the right, affinity goes down. This means hemoglobin is not as attracted to oxygen, and so it lets go of it quicker and easier. So if a left shift makes it easier to load oxygen, a right shift makes it easier to unload oxygen. Again, I would pause here and think about this until you understand this shift. So if the curve shifts to the left, hemoglobin's affinity for oxygen goes up. Hemoglobin's affinity goes up, it makes it easier to load. If the curve shifts to the right, hemoglobin's affinity for oxygen goes down. It doesn't like it as much. It lets go of it faster, which means it's easier to unload oxygen. So just by shifting this curve left or right, we can make it easier to pick up oxygen, or we can make it easier to drop off oxygen. Let's talk about the things that can make it shift one way or the other. But again, if we shift to the left, affinity goes up. It's easier to load. If we shift to the right, affinity goes down. It's easier to unload. Well, the things that cause this shifting are things like temperature, carbon dioxide, pH, and something called BPG, which stands for bisphosphoglycerate. So in other words, just changing temperature will shift the curve. Changing pH will shift the curve. Changing the amount of carbon dioxide will shift the curve, and then so will changing the amount of BPG. Let's look at these. Let's look at temperature first. So if we change the temperature, the curve will shift. If we increase the temperature, the curve will shift to the right. Remember what happens if you shift to the right. Affinity goes down, and oxygen is not as attracted to hemoglobin, and so oxygen comes off. It makes it easier to unload. Well, where are tissues warmer? They're warmer where they're active. So tissues like skin and muscle and heart, and those are very active, and they're much warmer than lungs. So when the blood gets to those tissues, the curve shifts to the right. Again, hemoglobin goes down, it's easier to unload oxygen. And we can see it in this graph. So the black line is normal blood temperature, but if we shift the curve to the right, look, we see the red line. It shifts way over. Hemoglobin's affinity is lower. It does not attract oxygen as quickly, and it drops it off faster. If we shift the curve to the left, the opposite thing happens. Affinity goes up. So lungs, which are cooler than tissues, curve shifts to the left. When the curve shifts to the left, hemoglobin's affinity for oxygen goes up, and it's easier to load oxygen. So, lungs are cooler, curve shifts to the left, easier to load. Tissues are warmer, curve shifts to the right, easier to unload. So without changing anything, we can make it easier to load and easier to unload. Also, the more active a tissue is, the further the curve shifts to the right. So tissues that are working hard get very warm. And when blood gets there, the affinity drops even more. And so even more oxygen is dropped off. It's also dose dependent. So again, the more you change the temperature, the further the curve shifts. Another thing that can change this affinity and shift the curve is carbon dioxide and pH. Well, carbon dioxide and pH are connected to each other because when carbon dioxide is added to water, some of that carbon dioxide will chemically combine with water. So carbon dioxide plus water will give us something called carbonic acid. Carbonic acid, like all acids, will give up hydrogen ions. So a hydrogen ion will come off, leaving what's called a bicarbonate ion. Hydrogen ions, bicarbonate ions will be in equal amounts. Well, hydrogen ions can be measured. They're measured by something called pH. And the more hydrogen ions there are, the lower the pH. So if I increase carbon dioxide, remember it's going to combine with water, give me carbonic acid, that's going to dissociate, and the hydrogen ions are going to go up. When hydrogen ions go up, pH goes down. These are very, very tightly connected. The opposite is also true. If I decrease carbon dioxide, less of it will combine with water. Less carbonic acid will be produced, and fewer hydrogen ions will be produced. Fewer hydrogen ions means an increase in pH. So this relationship is extremely important if you're going to stay in healthcare. It's something that you should probably just memorize. You shouldn't have to think about it. It's just something you should know. You'll use it again and again and again in all aspects of medicine. So let's go over it again one more time. Carbon dioxide plus water will give you this carbonic acid. Carbonic acid will dissociate, separate into a hydrogen ion and a bicarbonate ion. The more hydrogen ions you have, the lower the pH. So if I increase carbon dioxide, automatically I will increase the hydrogen ion concentration, and automatically I will decrease the pH. If I decrease carbon dioxide concentration, remember that there's going to be less carbonic acid. Automatically, I'm going to decrease the number of hydrogen ions, and automatically, I'm going to increase the pH. These three are always connected to each other because of this equation. They're so connected to each other that when we produce a graph, we don't need separate graphs. We use the same graph because they're exactly the same. If I increase the carbon dioxide, or I increase the hydrogen ion, or I decrease the pH, the curve shifts to the right. And if the curve shifts to the right, affinity goes down, and it's easier to unload. So if I look at tissues, they don't have the same amount of carbon dioxide. Tissues which are producing carbon dioxide have more of it. If they have more carbon dioxide, they have more hydrogen ions, and they have a lower pH. And so their curve will shift to the right. By the same token, if I decrease the carbon dioxide or decrease hydrogen ions or increase pH, the curve shifts to the left. This makes the affinity go up, and it makes it easier to load. This is what happens in the lungs. We're blowing carbon dioxide off in the lungs, so lungs have less carbon dioxide, less hydrogen ions, and a higher pH. So the curve shifts back and forth as we move through the circulatory system. In the lungs, the curve shifts to the left. In the tissues, the curve shifts to the right. So in the lungs, when the curve shifts to the left, affinity goes up, it's easier to load. In the tissues, when the curve shifts to the right, affinity goes down, easier to unload. So this is a graph of carbon dioxide. But look, this is a graph of pH. They are exactly the same graph. You cannot separate pH and carbon dioxide. So there's no need to have a separate graph. We can put them on the same graph. And so if you look at this, you can see normal arterial carbon dioxide, remember, has a partial pressure of about 40. If we change that, we can shift the curve to the left or the right. Also think about this. Tissues that are more active are producing more carbon dioxide. More carbon dioxide, more hydrogen ions, a lower pH. The curve is going to shift further to the right. Just like temperature, it's dose dependent. The more carbon dioxide there is, the further the graph shifts. So just by the reason that oxygen is lower, carbon dioxide is lower in the lungs and carbon dioxide is higher in the tissues, we're going to shift the curve back and forth, back and forth. So temperature has an effect, Carbon dioxide has an effect. Hydrogen ion has an effect. pH has an effect. There's one other thing that has an effect, and that's called BPG. BPG is a byproduct. It's a byproduct of anaerobic glycolysis. We only do anaerobic glycolysis when oxygen levels are low. You're running out of oxygen. So we begin to split glucose and we get the byproduct of BPG. The more BPG there is, the curve shifts to the right. And as it shifts to the right, it makes it easier to unload. So we had low oxygen, we're going to unload more to bring it back up. When you look at this graph, it shows this. Now, BPG is also called 2,3-DPG. So that's what we see here. So look, the green line is normal DPG, and you can see where the graph is. But if we have more DPG, we shift to the orange line. We shift to the right. Affinity goes down. Easier to unload oxygen. If we have less DPG, the curve shifts to the blue, to the left. Affinity goes up, and it's easier to load oxygen. So all these things affect that. That was oxygen transport. Let's look at carbon dioxide transport. Remember, carbon dioxide is more soluble in water, in plasma, than oxygen is. About 20 times more soluble. That means more of it is dissolved. Not 1.5%, but somewhere around 7 to 10% is dissolved in plasma. Another 20 to 23% will enter the red blood cell and attach to hemoglobin. But it does not bind to the iron. It binds to the amino acids. And so we call it carb-amino-hemoglobin. And then most of it, though, actually is transported as a bicarbonate ion. Well, we have to go back to that equation again. Remember, carbon dioxide in water, some of it will turn into carbonic acid, and that will dissociate into hydrogen ions and bicarbonate ions. It happens automatically, but it happens at a much higher rate if we have an enzyme for it. The enzyme is called carbonic anhydrase, and there's a lot of carbonic anhydrase inside our red blood cells. So carbon dioxide enters the red blood cell. There's carbonic anhydrase in there. So very quickly, carbon dioxide will combine with water to form carbonic acid, and then it will dissociate into a hydrogen ion and a bicarbonate ion. What happens is the hydrogen ion remains inside the red blood cell, but bicarbonate, as it builds up, will diffuse out of the red blood cell into the plasma. Well, if you look at this, carbon dioxide, when it came in, has no charge on it. But bicarbonate, when it leaves, it has a negative charge. This leaves positive charges inside the red blood cell. Those positive charges, when they build up, will create a potential, like we saw in the nervous system. Red blood cells don't need a potential. In fact, it interferes with their ability to do what they're supposed to do. So there's something that happens to counteract this buildup of positive charges. For every bicarbonate that leaves, a chloride enters. This is called the chloride shift. So in the tissues, carbon dioxide comes in, bicarbonate leaves, and for every bicarbonate that leaves a red blood cell, a chloride enters. Remember, some carbon dioxide travels just dissolved in the plasma. And some carbon dioxide goes into the red blood cell and attaches to hemoglobin and becomes carbaminohemoglobin. This is what happens during internal respiration. The blood is then going to go to the lungs, and the opposite thing is going to happen. So the carbon dioxide that was dissolved will leave the blood, enter the alveolus. The carbon dioxide that was attached to the hemoglobin will detach, leave the blood, and enter the alveolus. The other, though, is a little more complicated because the bicarbonate, remember, is in the plasma. So for it to reverse, first the bicarbonate has to re-enter the red blood cell, has to combine with that hydrogen that was in there, form carbonic acid, and then that, in the presence of the enzyme, separates into carbon dioxide and water. And then, and only then, can the carbon dioxide enter the alveolus. We also have the opposite problem we had before. Bicarbonate has a charge on it. When it comes in, it brings a negative charge. Carbon dioxide has no charge. So every time the bicarbonate comes in, negatives come with it, and it builds up inside the red blood cell. Again, we would get a potential. Well, just like a chloride entered before, now that chloride has to leave. So in the lungs, we have a reverse chloride shift. For every bicarbonate that enters, a chloride leaves. And then it's back to the tissues again. In the tissues, carbon dioxide comes from the tissues, enters the blood. Some dissolves. Some enters the red blood cell, attaches to hemoglobin. Some enters the red blood cell in the presence of carbonic acid, combines with water, forms in the presence of carbonic anhydrase, Carbon dioxide adds to water, forms carbonic acid, separates into bicarbonate and hydrogen ions. Bicarbonate leaves. For every bicarbonate that leaves, a chloride enters. And then it's off to the lungs. Carbon dioxide in the plasma diffuses out. The carbon dioxide that was attached to the hemoglobin separates. And then again, the bicarbonate is in the plasma. It has to re-enter the blood, combine with the hydrogen, form carbonic acid, and in the presence of carbonic anhydrase, separate into carbon dioxide and water, and then the carbon dioxide can leave. Again, for every bicarbonate that enters a red blood cell, a chloride leaves. So we have this chloride shift. So there's a chloride shift in the lungs, or in the tissues, a reverse chloride shift in the lungs. Chloride shift in the tissues, a reverse chloride shift in the lungs, over and over and over and over. And remember, this is internal respiration followed by external respiration. Internal respiration followed by external respiration. Well, we don't breathe constantly. People take a breath, they pause. They take a breath, they pause. The average person breathes about 12 to 15 times a minute. It's usually a 3-second inspiration followed by a 2-second expiration. Well, something is controlling that. What's controlling it is the brain, specifically two parts of the brain called the pons and the medulla. There are neurons there, and those neurons fire periodically and cause the muscles, like the diaphragm, to contract. Well, there are two different types of centers that are involved. In the medulla are what's called the basic respiratory centers. There's what's called the dorsal respiratory group which is mostly inspiration, and the ventral respiratory group, which is mostly expiration. So, inspiration followed by expiration. Inspiration followed by expiration. And it happens over and over again. Again, about 12 to 15 times a minute. We also always move about 500 milliliters of air when we're at rest. So 12 times 500 is 6,000. 6 liters of air move in and out of our lungs every minute. But sometimes we need more air. Sometimes we might need less air. When we need more air, there's a center up in the pons, which is called the apneustic center. P-N-E-U means air. When you put A in front of the word, it means no air. So this is the no air center. In other words, we don't have enough air. So we need to either breathe faster or we need to breathe more deeply. So the apneustic center says to those other two, breathe faster, breathe deeper. So we get more air. The exact opposite of that is called the pneumotaxic center. Again, P-N-E-U means air. Taxi means to move. So in other words, we're moving air, moving air. We're moving too much air. So the pneumotaxic center says, you're moving too much air, slow down. So it tells the centers in the medulla, don't breathe as quickly, don't breathe as deeply. So these two centers have influences over the basic centers. So again, we have this dorsal respiratory group. It's mostly about inspiration. We have the ventral respiratory group, mostly about expiration. And then we have the two that adjust respiration, the apneustic and the pneumotaxic. They influence and modify respirations. They also help us to smooth out the transition between inspiration and expiration. Again, apneustic increases respiration. Pneumotaxic decreases respiration. We also need to have some idea why we need more air, why we need to breathe faster, why we need to breathe slower. And so we have to monitor things. And the main thing we monitor is carbon dioxide levels. But remember, carbon dioxide combines with water. So when we have more carbon dioxide, automatically we have more hydrogen ions, and automatically we have a lower pH. If any of those things change, we monitor it. There are chemoreceptors that monitor the levels of carbon dioxide, hydrogen ions, and pH. So if carbon dioxide levels go up, we need to breathe more. We need to get rid of this carbon dioxide. And so respiration increases. If carbon dioxide levels fall, it means we need to breathe less. And so we adjust our respiration. So we're constantly monitoring this. The chemoreceptors are found in two places. They're found in the aortic arch, and they're found where the carotids divide. And then they're also found in the brain, in the medulla, where they monitor the carbon dioxide level of cerebrospinal fluid. Again, if carbon dioxide levels go up, or hydrogen levels go up, or pH goes down, we will increase respiration. If carbon dioxide levels go down, or hydrogen ion levels go down, or pH goes up, we will decrease respiration. Oxygen is also important, and so we also monitor oxygen levels. We have these same chemoreceptors in the aorta and in the carotid, and they detect oxygen. When oxygen levels fall, respiratory centers are stimulated, and in ventilation and respiration goes up. But it takes a fairly large drop in oxygen in order to stimulate increased respiration. This picture shows you where those receptors are. So again, you can see them in the arch of the aorta, and you can see them where the carotids split. There are other things that affect respiration too. We don't want to overinflate the lungs, so there's stretch receptors in the lungs to prevent that. Certain things in our respiration system will cause us to sneeze or cause us to cough. Those are just respirations. So if you get pepper in your nose, it's an irritant, and very quickly you'll change your respiratory pattern. You'll sneeze. We also have receptors in our joints and muscles to indicate when we start exercising. We're going to need more oxygen. Respiration goes up. We can also choose to breathe or choose not to breathe. You can hold your breath if you want to. You can pant if you want to. So there's voluntary control over breathing. And then sometimes things like pain or emotions can also change respiration. Some things can go wrong with the respiratory system. One thing is asthma. So asthma is when you have bronchospasms, bronchoconstriction, and it can cause by lots of different things. This is what that looks like. You can see the huge difference in the amount of air that could flow through there. Another thing is tuberculosis. That's a disease caused by a mycobacterium. Here's the mycobacterium, and it creates cysts or scars in the lungs. Sometimes these rupture and bleeding occurs. These are easy to visualize on an x-ray. Other things are things like bronchitis, emphysema. All of these things can affect respiration. And then there's lung cancer. Lung cancer is one of the leading causes of cancer deaths in North America. And almost all lung cancers are caused by smoking. So if you look at what smoking does to the lungs, you can see huge differences. There are three most common types of lung cancer: squamous cell, adenocarcinoma, and small cell carcinoma.